All right, let's go over the first question in the biochem section for full length 5, and it says, the association observed experimentally between the expression of miRNAs and the mRNAs in AR kidney transplants indicate that miRNAs regulate the expression of genes implicated in which types of immune responses. All right, so let's go take a look at what the passage stated. Let's look at experiment 2. So they're asking us to look at experiment 2. And it says right here that they measure CD3 levels, or CD3 mRNA levels, and CD20 mRNA levels. So the CD3 mRNA levels are giving us an indication of how much T cell expression there is, and the CD20 mRNA levels are giving us an indication of how much B cell there is in the AR biopsy specimen. And they found that there's a positive correlation between basically T cell and B cell expression levels and the levels of these three miRNAs, but not these three miRNAs. So it seems to me that these miRNAs are going to be controlling somehow the T cell and B cell expression levels. So let's take a look at this question. And it's essentially kind of a discrete here. You have to know what the B cell and T cell types of immunities are. And so we have adaptive immunity, which includes both. So let's say adaptive immunity. These guys include both B cell and T cell immunity, okay? And B cell is actually also commonly known as humoral immunity. The antibodies, it's an antibody-mediated immunity. And T cells are known as cell-mediated immunity, okay? Well, we know that it can't be B because it's not just B cells. We saw there was a positive correlation with T cells as well. Now, C says innate and cell-mediated immunity only. So we know that cell-mediated immunity is involved, but there's nothing about innate immunity. And what is innate immunity? Innate immunity are things like mucus or the hairs in your nose that catch germs as they come in. That's innate immunity, and they don't involve the B cells and T cells. So we don't ever talk about innate immunity, so we know it can't be C. And finally, D says adaptive and innate immunity only. So this would be all kinds of immunity. And specifically, we only talk about adaptive immunity and not innate immunity in that paragraph here in experiment 2. So we know it can't be D either, leaving us with A, adaptive immunity only. All right, question 2 says, based on figure 1, which patient is least likely to benefit from therapy with immunosuppressors? A patient whose transplanted kidneys are associated with miRNA expression patterns characterized by blank. All right, so first we have to think about what are immunosuppressants and what are they used for. Immunosuppressors are often used for people with transplants, and especially if they have rejections to the organs that they have transplanted or that they have transplanted inside of them. So we're going to be focusing specifically on the figure 1 acute rejection grafts here, because acute rejection grafts are the patients that are going to most need these immunosuppressors to better accept those kidney transplants. All right, so let's take a look. Characterized by high levels of MIR155. So high levels of MIR155 relative to a normal transplant, we see that high levels of MIR155 are characterized for patients with acute rejection of the kidneys. And so immunosuppressors would help these guys. So since we're looking for least likely to benefit from therapy with immunosuppressors, and patients with high levels of MIR155 will most likely benefit from therapy, we know it can't be A. B says low levels of LET7C. Again, we see that compared to normal transplants, acute rejection patients have low levels of LET7C, so they would benefit from immunosuppressors. High levels of MIR30A3P. So high levels MIR30A3P. Now here we see that high levels are associated with those with a normal transplant. So giving these guys immunosuppressants won't be as helpful as if we gave these guys to patients with or as if we gave the immunosuppressants to patients with acute rejection. So C is looking like the answer to me so far. But let's take a look at D. Remember to go through all of the answer choices. Low levels of MIR10B, so MIR10B, and we see that these guys, low levels are associated with people with acute rejection, which means they would benefit from the immunosuppressors. So we know it can't be D. So given all we have, C is the correct answer for this question. All right, let's take a look at question 3 of this passage. It says a graph of miRNA plus RNA denaturation versus melting temperature is shown. The arrow in the graph indicates the curve representing the melting temperature for which miRNA, assuming 100% base complementarity with its target sequence. So let's take a look at the graph real quick. Oops. There we go. If we take a look at the graph, we'll notice that the curve that this arrow is pointing at is all the way to the right. What does this mean? It means that it needs the highest temperature for it to denature just halfway. So if we look at the first graph on the left here, it requires a lot lower of a temperature to denature to a 50%. So what this tells us is that the miRNA that we're looking for is something that's going to be very stable. It's something that requires a lot of heat or high heat and increased temperature in order for that miRNA to come off of that RNA. And so how can we tell? So let's take a look at the passage, and what do we notice? We get sequences. And what are we looking for in these sequences? We're going to be looking for the sequences with the most amount of G's and the most amount of C's. And why is that? Remember guys, GC base pairs, they have 3 hydrogen bonds. AT base pairs have 2 hydrogen bonds. So what that tells us is that the GC base pairs are going to be stronger, and thus the sequences with more G's or more C's are going to be the binding, right? The binding between the miRNA and the RNA, that binding is going to be stronger if there's more G's and C's. Again, so if this is a miRNA and it's going to latch onto a target RNA sequence, okay? If there's more G's and C's right here, that strength of the binding is going to be stronger, and thus the temperature required to pull these 2 strands apart is going to be higher, and we want high temperatures here, so we want high G's and C's. So let's take a look. The first answer choice says MIR 142.5P. So this is the first one right here. Let's see how many G's and C's we have. 1, 2, 3, 4, 5, 6, 7. All right, we have 7 here. Second one is MIR 223. 1, 2, 3, 4, 5, 6, 7, 8, 9. MIR 10B. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And finally, MIR 30A3P. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So since MIR 30AP or 30A3P has the most G's and most C's, we know that this is going to be the miRNA that they're pointing at for this curve right here, making D our correct answer. All right, so this is the last question of the first passage. And it says individual nucleotides within the backbone of the regulatory RNAs discussed in the passage are held together by blank. So regulatory RNAs, they just mean RNAs, normal RNAs here. And because I can't draw, let's take a look at this pre-made photo of RNA. It's asking us, okay, let's take a look at these nucleotides. So this is a nucleotide, right? And this is another nucleotide, and so on and so forth. And so what are these nucleotides held together by? Well, notice that their bond kind of exists right here, right? And so these phosphate groups are what hold these nucleotides together. And specifically, these phosphate groups, these linkages are called phosphodiester bonds. So let's think about that. Let's break that down a little bit. So let's think about a normal ester. So this is a carbon-based ester, right? And so this is an ester. Remember, you have to have a carbon, you have the carbonyl group here, and then you have the oxygen that is attached to another group that's not hydrogen. So that's what an ester is. So why would this be called a phosphodiester? First, it's a phospho, so it's a phosphate instead of a carbon. That's the phospho part. And the diester, so let's see, where do we see the diester? So we know that we see that carbonyl here. We see the first oxygen that's attached to something that's not a hydrogen, and there's another oxygen here that's attached to something that's not oxygen. So that's what the phosphodiester means. So we'll see that the phosphodiester linkages are what's holding together these nucleotides, making B the correct answer. And let's just briefly go over the other answers here. It says disulfide bridges, so these are normally for polypeptide chains, right? So these are amino acids, proteins, not really nucleotides, RNAs, or DNAs. And we don't even see any sulfurs to begin with on any of these nucleotides, so it can't be disulfide bridges. Hydrogen bonds, so hydrogen bonds are non-covalent, okay? And these are all covalent linkages that we see here. And remember, hydrogen bonds, that's when you have a hydrogen that's attached to an F, O, or an N. So one of these has to be attached to a hydrogen, and then you have some attraction to another F, O, or N due to electronegativity differences. But again, this attraction is non-covalent, and what we're seeing here are covalent bonds, so it can't be C. Finally, glycosidic linkages. So this might be the closest answer, the second closest answer, but we'll go into why it's not quite exactly what it is. So what are glycosidic linkages? Glycosidic linkages are when you have, so let's draw the backbone of a sugar here. So this is the backbone. So let's say this is our sugar here, and we're actually missing a bunch of hydroxyl groups, but here, I'll just draw it in for the sake of being accurate. But I believe this is glucose, what I've drawn here. So this is our single carbohydrate. What are glycosidic linkages? Glycosidic linkages are when you have this anomeric oxygen, and this anomeric oxygen has to be attached to, oops, I'm gonna try to delete this hydrogen here. And this anomeric oxygen has to be attached to something that's not another hydrogen, right? So it can just be any other group. Now usually this takes form of being another carbohydrate, but it doesn't have to be a carbohydrate. Now the AAMC explanation does say glycosidic linkages attach different carbohydrates together, but from what I've seen, it doesn't necessarily have to be a carbohydrate. It just has to be something that's something else that's not a hydrogen. And so once this happens, this is when you have a glycosidic linkage. So this bond right here that they're talking about, that's a glycosidic linkage. But yeah, we don't see anything like that here. We don't have an anomeric oxygen. That anomeric oxygen is actually replaced by a nitrogen, and so that's not essential. And again, even if this was an oxygen, somehow this isn't what's holding the nucleotides together. So it can't be glycosidic linkages for those two reasons, making D incorrect and B the correct answer.